Hello, and welcome to Celiac.com news. Subscribe to our channel to get the latest celiac disease and gluten-free diet news. And if you have any questions, be sure to join the Celiac.com forum online at Celiac.com. And now, let's move on to more Celiac.com news. Breakthrough study on gluten-dependent activation of T cells offers hope for celiac disease treatment. Celiac disease is an autoimmune disorder in which the immune system launches an aggressive response against gluten, a protein found in wheat, barley, and rye. This response damages the intestinal lining, leading to debilitating symptoms and requiring lifelong adherence to a strict gluten-free diet. However, groundbreaking research recently published in Gastroenterology unveils new insights into how the immune response to gluten begins, potentially paving the way for innovative therapies, and perhaps even a cure. Understanding celiac disease and the role of gluten. For individuals with celiac disease, consuming gluten triggers an immune response that damages the intestinal epithelial cells, IECs, which line the upper intestine. This damage can cause severe gastrointestinal symptoms, nutrient malabsorption, and a cascade of systemic effects. The key culprit in this immune reaction has long been identified as CD4 plus T cells, specialized immune cells activated when gluten-derived antigens are presented to them by molecules known as MHC class II proteins. Until now, researchers believed that antigen-presenting cells, such as dendritic cells and macrophages, were the primary facilitators of this process. However, the new study has identified IECs themselves as crucial players in gluten antigen presentation and subsequent T cell activation. The discovery: epithelial cells take center stage. The study demonstrates that intestinal epithelial cells expressing human MHC class II proteins, HLA-DQ2.5, play a pivotal role in activating gluten-specific CD4 plus T cells. This process was observed both in patients with active celiac disease and in a specialized mouse model genetically engineered to express the human MHC class II molecule. Researchers have uncovered significant insights into how celiac disease develops and triggers immune responses. They found that intestinal epithelial cells in both humans with active celiac disease and gluten-immunized mice express MHCII molecules, which are critical for presenting gluten-derived peptides to the immune system. This expression increased dramatically when the cells were exposed to interferon gamma, a pro-inflammatory cytokine. Additionally, gluten exposure in lab settings triggered robust activation of CD4 plus T cells, a type of immune cell. This activation was characterized by heightened cell proliferation, increased markers of immune activity, and the release of inflammatory cytokines, including interleukin-2, interferon gamma, and interleukin-15. Furthermore, the study highlighted the role of bacteria in amplifying this immune response. Gluten digested by elastase-producing bacteria, specifically Pseudomonas aeruginosa, intensified CD4 plus T cell activation, whereas this effect disappeared when a mutant bacterial strain without elastase was used. These findings provide critical insights into how dietary gluten and certain gut bacteria contribute to the immune activation seen in celiac disease. These findings highlight the complexity of the immune response in celiac disease and the previously underestimated role of IECs in driving the process. Implications for celiac disease treatment. The discovery of epithelial cells' role in gluten antigen presentation opens new doors for therapeutic strategies. By targeting the mechanisms within IECs that facilitate T cell activation, researchers could potentially disrupt the immune cascade at its source. Emerging research suggests promising potential treatments for celiac disease aimed at disrupting key processes in immune activation. One approach focuses on inhibiting MHCII expression in intestinal epithelial cells, which could block gluten antigens from being presented to CD4 plus T cells. Another strategy targets pro-inflammatory signals, such as interferon gamma, to prevent the upregulation of MHCII and reduce immune system overactivation. Additionally, modulating the gut microbiota shows potential, as certain bacterial enzymes, like elastase, enhance gluten antigen processing. Adjusting the microbiome's composition could help minimize this effect. While these treatment options are still in the early stages of development, they hold promise for advancing toward disease-modifying therapies for celiac disease. A step closer to a cure. Currently, the only treatment for celiac disease is a strict, lifelong gluten-free diet. This diet is challenging to maintain and does not completely eliminate the risk of inadvertent gluten exposure, which can trigger severe symptoms. The study's findings offer hope for a future where treatments go beyond symptom management to address the underlying cause of the disease. By pinpointing the initial stages of the immune response to gluten, researchers have provided a blueprint for interventions that could potentially prevent or reverse the condition. While a cure remains on the horizon, the knowledge gained from this research brings it closer to reality. A new era in celiac disease research. This groundbreaking study sheds light on the critical role of intestinal epithelial cells in celiac disease pathogenesis, reshaping our understanding of how gluten triggers the immune response. The findings not only deepen scientific insight, but also offer practical pathways for developing innovative treatments. For the millions of people living with celiac disease, this research represents a beacon of hope, an indication that one day, the burden of a gluten-free life may be replaced with the freedom to enjoy food without fear. As researchers continue to build on these findings, the dream of a cure for celiac disease feels more attainable than ever. To get the latest celiac disease news and information, be sure to subscribe to celiac.com news. Thank you for tuning in, and see you next time.